In this electrolysis cell, we're going to be producing an electrochemical process that involves a non-spontaneous reaction to produce a product. The electrolysis in this was originally produced by Michael Faraday, and he produced Faraday's constant, which involves the charge of the coulombs per mole per electron. So in order for this experiment, first you're going to take 2 copper pieces. They need to be about 1 inch, and you need to sand them off with sandpaper in order to get any oxidation off. Then you need to measure the mass before you place them in your beaker. Next, what you're going to need is you're going to need a voltaic meter in order to measure the amount of volts produced, and a battery supply. Then you're going to need to attach your 2 pieces with the voltaic cell. You're going to have the pointy edge, and you're going to need to attach it with the alligator clip. That will allow it to be measured securely. Make sure that the 2 ends are not facing each other or touching. They're facing opposite and not touching, so that no reactions occur. Next, you're going to need a solution that involves the copper and nitrate in order to allow the ions to transfer from one cathode to the anode. Next, you're going to turn it on for a minute of 20 seconds, and every minute you need to record the amplitude in order for your data analysis. After your 20 minutes has passed, turn the voltaic cell off, take off the amps, rinse them in alcohol and in water, and then you're going to measure them in mass, and then you're finished.
So this is a galvanic cell, and these are the 2 half reactions. So this is the copper anode, and this is the lead anode. And these solutions are copper nitrate and lead nitrate, and they're connected by a salt bridge that allows the ions to flow through to each half cell. For each trial of the lab, you would use the voltmeter to measure the voltage, and then once each trial is done, you would replace the anode, you would replace the anode and the cathode. So for example, this is copper and lead. So for the second trial, it would be copper and zinc, and for the third trial, it would be lead and zinc. So you're examining each of these different galvanic cells in the lab, and that's it.